I try to get ahead now because I make, so now I got my, let's see if I can go ahead. Okay, good, fantastic. It's not going ahead. So, let me see. Okay, very, um, recently, I don't know why it's going so badly, but try. Checking for updates. Oh, come on. Okay. Try again, eh? I don't know why it's not getting, it's not going ahead. Strange stuff, but we try. Now it should be okay. I use this one is better. So, as you know, recently, last year, basically, a few months ago, the repertoire of the mutation, of cancer mutation, has been better precised, I can say. And for many of the human tumors, a better definition of all these human tumors here, a better definition of what the significant mutations are has been achieved. And even not all of these mutations are really driving genes, but some of them are. So we got a more precise idea of what the gene landscape of human tumors are at the moment. Despite that, a substantial number has been described, but the mechanism that actually are driven by this mutation is still just partially known. And now it's time to start from this, can I say, baseline. So what the mutations are, to understand much better what the effects of these mutations are and how this mutation can be exploited in terms of therapeutic um, interventions. So all hands on deck, because it's time to, actually we got a lot of work to do starting from here, but now I think that even the more difficult part of the job probably is starting. This is I can, let me say, let me call it a kind of cautionary paper published by Vinay Prasad a few days ago, a couple of days ago, a week, in which he demonstrates very clearly, Vinay Prasad is a very smart guy, in my own opinion. Is a very critical and a very high capability of analysis. And this shows, as you can see here, that less of 8% of patients with advanced disease at the moment can really benefit of target therapy in terms of immunotherapy, tyrosine kinase receptor inhibitor, or all the other type of treatment we can figure out at the moment. So despite this big hype we are deluged with every day, the real impact of these new treatments are still very, how can I say, still a bit murky and not very clear. Probably because maybe some of these treatments are a bit emphasized too much in because of many factors. And probably another main reason is probably we don't know exactly what the biological effects of these targets are. So it's a caveat, because we have to avoid definitely our cautionary tales. I don't know if who of you recalls the story with anti-angiogenic. So anti-angiogenic has been hailed like a kind of definitely conclusive tumor therapy. So with anti-angiogenics, everything was curable. And after more than 25 years, and 25 years, the bottom line of the use of this anti-angiogenic is just something that can be useful sometimes in some tumors in combo therapy with our drugs, but for sure the role of this class of drugs is significantly reduced compared to expectation. And the guy who won he never won basically the Nobel Prize. He discovered the anti-angiogenic agent. Said, okay, he was a very smart person. He said, and very sense of humor. And he said, okay, if you are a rat and you got the cancer, it's likely that I'm gonna cure you. And probably was very right, at least partially right. That said, if we are talking about mesothelioma and we start thinking about what mesothelioma in terms of mutation can offer us for treatment, for sure, the two most relevant genes we can figure out to be druggable, whose mutation can be druggable are CDKN2A, so RB pathway and cell cycle, and BAP1. BAP1 is mutated in a significant number. CDK2A is mutated in almost 60%, and also malignant mesothelioma in malignant mesothelioma, and also BAP1 in the same tumor with different percentage of mutation is frequently detected mutated in epithelial subtype, as you very well know. In renal cell carcinoma, BAP1 has been considered a kind of landmark or kind of litmus test to split the renal cell carcinoma patient to big subpopulation, sensitive to different drug and with different prognosis. And that's what we are trying to do with BAP1 and meso. At the moment, as you know, there are some germline mutation, and there are some somatic mutation. Okay, up to 60% of somatic mutation are detectable in tumor in meso cells, in mesothelioma cells. Moreover, it's very easy to detect BAP1 mutation because usually is universally accepted that the cytoplasmic staining of BAP1 means mutation. So when the protein is not expressed at all, or the expresses in cytoplasm, means that this protein is not synthesized at all, or otherwise is inactivated, so cannot work and cannot protect cells from DNA damage, for example. I have to use the arrow, otherwise. As you know, BAP1, other than being involved in DNA damage, as you know, mesothelial normal cells with BAP1 mutation are not able to repair DNA damage or asbestos induced or whatever induces DNA damage. And this means that damaged cells, DNA damaged cells can survive to apoptosis because BAP1 is a gatekeeper for calcium influx, as you know. So less calcium, BAP1 mutated, less calcium, less apoptosis, more mutated, DNA mutated mesothelial cell do survive more easily, is that this DNA damaged mesothelial cells surviving DNA damaged mesothelial cells do become cancer cells. Another big role of BAP1, as you know, is the capability to switch metabolism from a more oxidative metabolism, BAP1 wild type, to a more glycolytic metabolism, BAP1 mutant. this is being demonstrated in mesothelial cells. In mesothelioma cells, things are a bit more complicated, but basically, there are some papers of ours, a bit old now, 2013, and other results that are coming out soon, that actually go in the same direction. So, I mean, it seems that mesothelioma cells, white type mesocells are more oxidative, mesothelioma cells, not stem cells, and mutant mesothelioma cells are more glycolytic. So more oxidative if you are white type, more glycolytic if you are mutant. And this is very similar to what happens in mesothelial cells, normal. Basically, as you've seen here, this is a lot but one white type has got a quick glycolytic adaptation. So if you I'm gonna say starve these cells, they can go switch. So they are in hypoxia condition. They switch rapidly to glycolysis, but usually I've got as can you say a very poor reservoir of glycolytic capacity, and they do die. And the same thing is being demonstrated some years ago, as I said, back 2013. They can cope with damage. In this case, it's not hypoxia, it's oxidative phosphorylation. But in the end, they can cope for a while, but they can cope for a little while, but they cannot survive longer, and then they undergo apoptosis if they are white type, because are prevalently oxidative. Okay. I try to get ahead if I can. Now it does not accept my arrow. Now I accept my, okay, now I accept my. And so we can try and, I can say, draw some preliminary conclusions. BAP1 regulates calcium, as I mentioned before. So if BAP1 works regularly, it can allow calcium to enter the cell, mitochondrial cell, the cell mitochondria, sorry, can induce apoptosis of damaged mesothelial cells. And this because basically, as I mentioned before, is a calcium gatekeeper. So the more calcium is, more calcium influx you have got, the more likely is the cell can undergo apoptosis. Again, BAP1 mutant mesothelial rely on glucose, and but actually these are big epiphenomena of what happens. We still lack a deeper insight into what the mechanism involving this different use or better, this different metabolic profile is based on. But now, from, if you like, basic biology to clinical implication, what we are saying, otherwise we are all doctors, so we have to try to treat our patients. As you already know, BAP1 is a tumor suppressor gene, whatever, but what happens is that if it's considered a tumor suppressor gene, it is in mesothelial cells, there's no doubt that is a tumor suppressor gene. When your BAP1 is mutant, that's not working for some reason, it's not expressed, those got a mutation that actually is inactivating mutations. These patients, especially the germline mutation, but also the somatic mutation, these patients do survive longer. So the question is BAP1 really a tumor suppressor gene? Is really a gene that can promote apoptosis under stress? It can be oxidative stress, it can be hypoxia, it can be whatever you want, and is still a shield of against the mutated cells and against carcinogenesis process. As far as mesothelial cells, normal mesothelial cells is concerned, the answer is definitely yes. But as mentioned before, when we look at what happens in tumor cells, okay, this is bit is something I'm going to show you, but maybe to be quick, try to show you this. When you have a look, this is a this is an heads up, okay, confidential among us, okay. It's something that is coming out soon anyway. The intriguing thing here is that mesothelioma cells, mesothelioma, not mesothelia, that are BAP1 Y type, do generate, this one, do generate much less, sorry, these two cell lines are Y type, okay? If we silence BAP1 or IP3R3, sorry, IP3R3, I did not mention this actor, character of this comedy before. IP3R3 is a kind of gatekeeper on endoplasmic reticulum, and is the one that is activated by BAP1, and is the one that actually opens the calcium channels to allow calcium to enter the tumor or the normal cells. So if you silence BAP1, or if you silence downstream IP3R3, so basically, if you have let's say you obtain a mutant, BAP1 mutant cells, the capability of these cells to generate stem cells is basically zero. If you have a look at what happens in basal conditions, spherogenesis from mutant cells is usually much lower than the spherogenesis for from Y type cells. Okay, which is something here is missing, but just to let you know, on the baseline, paradoxically, and on the contrary of what expected, the so-called good BAP1 Y type do generate a lot of spheres, and the so-called bad BAP1 mutant mesothelioma cells do not generate, or very few spheres. But if you silence BAP1 in mutant cells, sorry, in Y type cells, BAP1 or IP3R3, the number of cells of spheres is almost zero. So it's very significant, very clear cut. This is very clear cut. So spheres, as you know, are cells that are basically we see no. Spheres are cancer promoters, if you like, usually are LDH positive, and there are very few of them, but spheres 0.1, 0.2 of all the tumor cells are stem cells, so I got stem cells markers. these spheres are usually enriched by tumors. So the percentage of stem cells in these spheres is significantly higher, is up to 1-2%, and most of them are LDH positive in mesothelioma stem cells. And but the capability of our tumor, of this tumor, to generate spheres is completely BAP1 IP53 dependent. So if you silence BAP1 spheres, the number of spheres drop. And so probably we try to, throw to try to draw some preliminary conclusion. It could be one of the mechanisms why we see this striking difference in terms of survival between BAP1 wild type and BAP1 mutant patients. So the take-home message here is that mesothelioma is BAP1, sorry, is a tumor suppressor gene in mesothelial cells, because basically via favoring apoptosis, but it seems all the data go in the same direction. It seems working like an oncogene in mesothelioma cells, because it promotes stemness, and it promotes stemness via p53 and calcium influx. There are two mechanisms underlying what we see. I just elaborate it up. One is that probably there's an interaction with P10, and because P10, BAP1 stabilizes AKT, AKT degrades P10, and this affects IP53 phosphorylation status. Another one is linked to FXP2, that is a protein that is a ubiquitin for IP53. But this is something, again, it's up. There's a more, so this is an epiphenomenon, what we see. The mechanism underlying this phenomenon probably is more complicated. We are close by, not still there, but getting closer. But this, in the end, that is the most important thing, is that is something that allowing us to distinguish two main subpopulations of mesothelioma patients with distinct prognosis and susceptibility to standard and novel therapy. Therapies. Clearly, all the drugs that are potentially able to interfere with the glycolytic metabolism are potential drugs for mutant. And the opposite, all the drugs that can impair the oxidative metabolism are potential candidates for wild type. This slide is a bit, is a bit old. This TMA analysis has already been done, and actually there is a good correlation between IP53 and survival. The more IP53 our patients express, and it seems that the shorter the survival is, because this mechanism is still working. But one IP53 type are still happily working together, and this is affecting spherogenesis and, in the end, our patient survival. Let me see what can I do again, because otherwise, okay. So IP53 can become a novel therapeutic target for mesothelioma. Honestly, we have to, we already try and do some drug docking. So what type of drug among the ones that has been shelved by FDA so far can have a physical chemical interaction with IP53. We spotted a couple of drugs that are potentially a good match for IP53, are still so we are still exploring if our hypothesis stands up. The other option is designing new small molecules that are specific for IP33. Clearly, IP33 is very ubiquitarious protein in human body, so the main concern here is toxicity, but we try and see what happens. Well, as I mentioned, when presented, actually my title is encompassing immune response against the mesothelioma, and this is the second big field of our activity at the moment. In collaboration with Dublin and Milan, we are working on something I'd like just to show you quickly tonight. First of all, it's still in press on Frontiers Pharmacology, a comment of ours, an opinion article, where we say, is there already a bit provocative question, if you like, but is there already need of a reckoning on cancer immunotherapy? I anticipate you that our answer is definitely yes. And for many reasons, this is a bit unusual opinion article because it's got almost 50 references. Because given that we were aware that this is a rather provocative message, and so we try to, how can I say, underpin our message as much as we can with a lot of references that actually support our statement. And more than ever, I think that for immunotherapy of mesothelioma and immunotherapy for cancer, thinking outside the box is absolutely paramount. One of the reasons why immune checkpoint inhibitors are effective sometimes, especially in some type of tumors like non-small cell lung cancer, melanoma, renal, head, neck. Difficult to find, really, despite all the hype, to find other tumors where immune checkpoint inhibitors really affect the survival of our patients, unless we are patient with performance status 0, or we are patient with very, very limited disease. But one of the reasons why probably is that tumors, stroma, is largely infiltrated by Tregs. Tregs, as you know, are CD4, 25, FOXP3 positive cells. They are deeply immunosuppressant. They release a lot of bad stuff like TGF-beta or IL-1 or CLX that deeply abrogate immune response. And these Tregs have been identified in many tumors, breast in this case. So how about Tregs in meso? Oh, first of all, immunohistochemistry shows us that there are a lot of, as you say, proliferating FOXP3 positive cells in mesothelioma. There's a massive infiltration. And this is not a good mark of response. Moreover, Tregs from patients with, usually, but particularly from patients with mesothelioma, circulating and from tumor tissue, are deeply immunosuppressive in terms of capability to suppress the proliferation of CD4 T cells from healthy donors, as you can see here. So mesothelioma is full of Tregs and significantly affects T4 proliferation. This is funny, if you like. It's not funny, but you know, I mean, it's intriguing, more than funny, because Other than this, mesothelioma cells are very good APC. We published a very old paper. I was still a young boy, full of hopes, many years ago, where we demonstrated that mesothelioma cells were pulsed with PPD or tetanotoxoid, was able to induce lymphocytes proliferation from lymphocytes taken from the same patients, So patient immunized for tetanus toxoid or PPD with mesothelioma, fortunately, we challenge his cells with his T cells, and these tumor cells were able to induce lymphocyte proliferation, but at the same time, they release a lot of TGF-beta, and these lymphocytes quickly, swiftly switched from a Th1 to a Th2 immunosuppressive profile. So Th1 interleukin-2 gamma interferon, and Th2 interleukin-4 and 10. So these cells are bad cells. They don't like immune response, and they try to abrogate immune response, and are different as well. So if you see, tumor infiltrating cells from meso are different from the same Treg cells from blood. So they are immunosuppressant and different, and they are changed by the tumor stroma. Probably the microenvironment or whatever is going to change these cells and make them even more immunosuppressant. This is a neat map. Is a neat map of Tregs, intratumor mesothelioma Tregs versus healthy donor. The heat map is different, significantly, as you see. And is even different if you compare infiltrating mesothelioma infiltrating Tregs versus non-small cell lung cancer infiltrating Tregs. Okay, the heat map of these Tregs is highly different compared to the Tregs infiltrating non-small cell lung cancer. I don't know what happened. Why? We don't know. But in the end, if you have a look at the genes of this heat map, this mesothelioma heat map includes much more immunosuppressant genes than Tregs from non-small cell lung cancer. So there is something that actually is more than a clue at the moment. And now I finish. The new heads up is something even more, is beyond the immuno checkpoints, okay? It's clear that this Treg stuff and this Treg's immunodepression or immunosuppression is likely to be stroma related. One of the most potent molecules stromal molecules that can drive immune response is at ATP, that, as you know, can be degraded to AMP, and then can be degraded to adenosine. So usually there are two ectonucleotides that can do this job. One is CD39 that transform ATP in AMP, and then CD73 that can transform AMP into adenosine. Adenosine is the culprit here. Adenosine is a very potent immunosuppressant, very potent. As you say here, is degraded by CD26. And by the way, as you know, overexpression of CD26 in mesothelioma is a very good prognostic factor for our patients. So adenosine is likely to be a culprit, to be responsible of something. And, try to go ahead, its effect is exerted via two types of receptors. One is called ADORA2AR receptor, and one is called A2BR receptor. So B receptor, basically. So adenosine binds to these receptors to exert its effect, but usually is via phosphocrep. And phosphocrep is constitutively expressed on mesothelioma cells. This mechanism has already been identified in renal cell carcinoma, and using antagonists for adenosine, so disallowing adenosine to bind to its natural ADORA2B or A receptor, is able to restore, to increase sensitivity to immune checkpoint inhibitors, or even to restore the sensitivity of renal cell carcinoma after this patient became resistant to immune checkpoint inhibitors. At the same time, this pathway affects patients with mesothelioma. In particular here, as you can see here, I got a lot, sorry, I forgot my phone on. And I was saying this receptor, ADORA2B, is strictly, I don't know if you can see it now, because it's covered by this. Okay, is strictly related to worse survival. So the more ADORA2B your patients have got, the shorter the survival is. Clear cut. Do our cells, mesocells, express these receptors? I can say yes. ADORA2B is mainly expressed by our tumor cells, mesocells. ADORA2A is not well expressed, or at least is not expressed as much as ADORA2B, which are better, many cell lines, you can see here. With just one exception that is very intriguing again, because just these are cells that have been selected for platinum resistance. And just cells with platinum resistance do express ADORA2A, mesocells express ADORA2A. What does it mean? I don't know. This is something that is under examination and under test. The same, so we had a look at our, the same happens for CD73. It seems so, at least the CGA data. We check our patient, small group so far, but we check if CD73, that is the ectonucleotidase, that is the last step transforming AMP into adenosine. And the more CD73 your patient, these are tissue, it's TMA again, the more CD73 your patient will express, the worse the survival is. So if they produce more adenosine, basically, they survive less. And this is not the important thing. How can I say? If we think on what we have said before on BAP1, has been basically that we can distinguish two main big subpopulation, and these two main big subpopulation have got BAP1 positive, BAP1 negative, better and worse survival respectively. And likely that this BAP1 status can offer us different druggable targets. In this case, we are a step forward, if you like. First of all, there are already drugs that can specifically address Tregs. Are in phase 1, early phases. with good toxicity, so very, very safe, and with some proof of activity still to be confirmed. But many of these, but there are already a battery of drugs that can be used to inhibit Tregs, or at least inhibit what Tregs, and this is something probably in the future, but when our heat map will be better defined, we probably are going to spot a lot of new targets selectively expressed by Tregs that can be druggable targets. So Tregs analysis on more patients, because the problem here that we have to analyze our Tregs from surgical samples. It is not very easy because, first of all, not all the patients, very few patients, unfortunately, are candidate to surgery, and we need fresh tissue, so it's very complicated stuff. But when we are hopefully not so, we won't so long, when we'll be discovering all the signals that induce Tregs immunosuppression, we'll be able to spot a lot of new drugs. We are even as, so what can I say, with adenosine, we are even more ahead, if you allow me this expression, because there are already drugs that are already being used in renal cell carcinoma. We're already in clinical trials in phase 2, early phases, and we are actively pushing a lot to start a new trial using different antagonists. One is for a 2A receptor, or anti-CD73, already available in clinical settings, even though very early phases, phase 2, not beyond. But this could be something that actually is going to change, if you like, the landscape, because using, for example, immune checkpoint inhibitor plus something directly affecting what the evidence suggests us, it is the mechanism underlying the resistance to immune checkpoint inhibitor, could be, if you allow me to say, a kind of master key to improve significantly the efficacy of this immune checkpoint in all the tumors, because it's not just a problem with mesothelioma. With mesothelioma, is a bigger problem, but with other tumors is not that far from mesothelioma at the end of the day, because the toxic microenvironment and the bad inflammasome, if you like. And so this is a problem that is across the range in oncology. And so if we can identify a combo therapy addressing stroma and tumor cells at the same time, we probably can really be to find a game changer for us. Just one word on this new the problem is, probably someone of you could argue easily, okay, but now why are you skeptical on immune checkpoint at the moment when the new frontline therapy for mesothelioma, it seems to be approved recently, is ipilimumab and nivolumab? Okay, if you think so, I invite you warmly to have a good look at our comment when it will be published, and to another paper will be coming out hopefully soon, where all the problem of this trial and many other trials are, I think, sufficiently underlined in terms of suboptimal control arms, patient stratification, and so on. Anyway, so the take-home message here is that there are many things brewing. Some of these things probably will be useful if used together. We don't have any magic wand. At the moment, unfortunately, our patient have got a survival that, give or take, is if they are lucky and they are lucky to be treated with chemotherapy plus bevacizumab, but at the moment is still the frontline therapy as for European guidelines, and at the moment still tops ipilimumab nivolumab. In this case, they are going to survive 3 more months than just with chemotherapy. And I think that all of you do agree with me that is not enough. Thank you very much. I finish. Sorry.
Thank you.
I stay within an hour, so it's fine. Eh? Okay. I check the clock. So it's fine.
Thank you, Luciano. There is a major problem in Turkey in terms of merging basic science and clinical science together. This problem is a big challenge on our scientific research. Today, we have listened to a very essential person regarding this issue. So your information that you shared with us was very beneficial and excited for us. Thank you. I hope clinicians in Turkey will start to take PhD degree on basic science, and some labs of basic science will be settled within our clinics. Also, I would like to thank to my colleagues who participated to this seminar, and I congratulate them to follow this complex topic. Now, so we move on to question and answer session. Will be coordinated by Dr. Güntülü. Güntülü?
Thank you. Thank you, Professor Mutti. It's a great lecture. We have a question from Akif Turna. He's a surgeon. He thanks you very much for the inspiring lecture. Did you stimulate tumor infiltrating T lymphocyte in culture and look at the activity and number of Treg cells again? If you did gene heat map change? And did you look at IL-17 gene expression?
So no, I start from the last question. No, I didn't have a look at the IL-17 yet. It's still something ongoing. The first question was, sorry?
The first question is, did you stimulate tumor infiltrating T lymphocyte in culture and look at the activity and number of Treg cells again? If you
stimulate with something, and okay.
Yes.
Okay. This is in terms of, you think a kind of, if you like, CAR T using Tregs. No, something can be done. Why not? It could be, I'm going to say, honestly, now these results are coming out. Once published, and further experiments are definitely, yes, could, because once we have a more precise map, gene map, genomic map, of the genes more likely to be involved in this immunosuppression, Tregs derived or dependent immunosuppression is like. At that time, we could be more precise in targeting this, the protein and the pathway affected by this gene. And probably we will be more likely to be successful to restore or to make these Tregs become a bit less depressant. So the thing is more important to make these cells more depressant, or is it more important to increase the efficacy of NK or other cells that are immunoactive? This is a good question in the end of the day, and probably, if this is a very tumor-specific approach, because what is clearly drives me crazy is that you cannot use the same drugs to restore immune response in all the tumors, because all the tumors are different. All the tumors are different stroma. I'm tempted to say that every patient has different tumor stroma. So unless you have got a more precise idea of what happens, if more immunosuppression or this decrease in immune reaction, if you like, is the main component of immunosuppression in the patient, we are not sure to work on the right engine. What can I say? Okay.
Okay, thank you. Is there any other questions from the audience? Dr. Mutti, you told us BEP1 could be a oncogene sometimes, right?
Yes.
When does BEP1 show tumor suppressor effect? When oncogene effect? How can we do? How can we know?
Basically, all our results go in that direction. So the clinical results show that white type patients have got a worse survival, and white type mutant have got better survival. This is clear cut, because our epidemiological study, so it's a couple of Meyer curves, so there's no doubt about that. The other big stuff is that actually why? This is a problem. So we think that actually the capability of BAP1 to determine spheres generation is one of the main reasons why this is this clinical effect is observed. And the fine mechanism of this increase or decrease pherogenesis is still under very active investigation. It's not has not been, can I say, has not been achieved enough so far. But at the moment, you know, we know that our BAP mutants are got slightly better prognosis, and that our white type got slightly worse prognosis. To emphasize, so I mean, I'm going to say to augment the biological machinery by which these two clinical effects are observed is our aim. I cannot answer your question yet. The preliminary data suggests the stemness generation is the key point between the two subtypes. The mechanism by which the stemness occurs is still largely unknown. For sure, some of these molecules, like IP53, are potentially a druggable target for patients, white type patients. But I can say, I cannot say more because we don't know more. This is our frontier, Adrian's wall.
Okay, thank you. We have got many thanks from our colleagues, Professor Mehmet Karadağ, Professor Ülke Yılmaz. We have one more question for you. Did you study the behavior of the other cellular components of stroma apart from T cells in mesothelioma?
It's a good question again. Clearly, M1, M2, no, we have not studied directly, but clearly M1, M2, for example, are very interesting subpopulations that are absolutely worth to be studied. And another big importance, relevant population are fibroblasts, cancer-associated fibroblasts, particularly F2, cancer-associated fibroblasts. There are paper very clear cut showing that inhibiting F2 protein in cancer-associated fibroblasts restores in vitro and in vivo models sensitivity to immune checkpoint inhibitors. And Tregs, among all the best things they do, is also that of stimulating cancer-associated fibroblast proliferation. So this basically leads to a kind of lymphocyte exclusion. So it's still, what happens is mesothelioma is an inflamed tumor. It's not desert, it's inflamed, but it's excluded. So all this stroma and fibroblasts and whatever disallow tumor cells to get closer to immune cells to go closer to tumor cells. And that's another reason why, for example, M2 are important for this, because they secrete a lot of immunodepressant cytokines, and fibroblasts because they exert a kind of mechanic effect. event, so disallow the interaction between immune cells and tumor cells.
Okay, thank you. Let me see the, yes, we have a thanks from Binur Önal. She thanks you so much for this multidisciplinary stimulating lecture.
I like what Muzaffer told us before. So I think to be a good doctor right now, probably now I'm going to retire in the next, I don't know how many years, but I don't have so many years as I had before ahead of me. But anyway, I think that the next generations of doctors should have a more precise idea what biology can give us, because otherwise there is a big discrepancy between clinicians and biologists. And until we won't be able to plug this gap, we won't be able to achieve, really to exploit all the knowledge we are getting in oncology, not just on mesothelioma, especially in mesothelioma, however. Okay, thank you very much.
Thank you.
So,
bye-bye.
Thank you, Luciano. I hope we can see ourselves again in good and healthy days.
again, yeah, when the virus will be finally dead, not ourselves. Okay.
Thank you. Good.
Thank you so much.
My pleasure. Ciao.
Arkadaşlarıma çok teşekkür ediyorum katılımları, destekleri için. Hepinize iyi akşamlar diliyorum. Görüşmek üzere.
İyi akşamlar.
Okay, ciao.
Yeah.
Bye bye.